Dr. Rhoades grew up in upstate New York and did graduate studies in both mycology and lichenology in Oregon, received an MS at Oregon State University and a PhD at the University of Oregon in the 1970s. From 1977 to 2009, Fred was an instructor of biology at Western Washington University in Bellingham, where he taught a variety of courses until retiring. He currently lives in Bellingham and continues to give occasional programs in mushroom and lichen forays. Fred continues to spend much of his field time photographing a variety of lower life forms, including mushrooms, lichens, bryophytes, slime molds, and the odd plant and animal. All right, so Dr. Fred, take it away.
Thank you very much, Laura. I'll share my screen here. Oh, wait a second. Not sure I did that right. Let me start again. There we go. So I thank you for inviting me. I didn't quite know what to expect. And Melissa invited me, so thank you, Melissa.
Yeah.
Fred, for being willing to do this presentation and to come down to Oak Harbor to do an inspection of what we have here.
Yeah, so I must say, living in Bellingham, you might be surprised to hear, we have very few Garry Oaks up here.
Mm-hmm.
We're right at the end of their distribution. There are a few here and there on some overlooking bluffs. There's one very odd tree that's quite old and very small in a local park. But other than that, it's not a common tree. So I was happy to come down and go out with Scott and Melissa, take a look at things down there. And of course, I did a lot of work down in Oregon. So I appreciate Garry Oak as a species. And I'm very, very happy to hear, well, first of all, I was surprised to hear that you're having, you're losing your oaks in, of all places, Oak Harbor, but happy to see that you're such a group to bring them back. And one of the things that I was thinking, I actually have quite a long presentation here. I don't know how much time I have, but I'll try to get through it and go through a lot of the slides very quickly. Maybe more than you really want to hear about lichens. But it's an interesting group of organisms. I'll tell you a little bit about them and why they're important ecologically and maybe less important to humans, but there are some interesting things that humans do with lichens. And then I'll go through some of the species that you're likely to see on your oaks or maybe in the future on other oaks in the area. Resources, I can send you a list of these of particular interest. I mean, these are if you want to get into identifying lichens. And I must say that if you are interested in looking at lichens or other small organisms like lichens, like the bryophytes on the oak trees, you really need a hand lens, the tool of the trade. There is a paper specifically about macrolichens in Garry Oaks in Washington. They didn't, John Villela and some others did this. And you can actually download this thing and send you the link. But they didn't really get up to Oak Harbor. They were up on the other side of the Puget Sound. I wrote a thing for the San Juan Islands, lichens of South Lopez Island, that you might still be able to find. A lot of the things that you would expect to find in Garry Oak are at least away from the marine influence, the same sorts of things that I found out there. And there are these other resources as well. So anyway, what are lichens? They are actually a combination of ingredients. They're a symbiosis between a fungus and some kind of photosynthetic party. The fungus we call the mycobiont, and the photosynthetic part is either an alga or a green alga or a blue-green bacterium. They build a structure called the thallus, and at the very beginning of trying to understand the different kinds, we divide the lichens up in between those that are leaf-like, the foliose lichens, those that have kind of a paint smear on rock or on wood, crustose lichens, and those that are little shrubs or dangling strands, which are the fruticose lichens. These thalli, which is the plural of thallus, love environments where it alternates between wet and dry. So the Pacific Northwest is really a perfect environment for lichens. There are probably several thousand lichens around in all of the Pacific Northwest. I found over 200 there over in South Lopez Island in that one tiny little location I was looking at. And likely, if you were to start looking at Garry Oaks around Oak Harbor, you might find upwards of several hundred. I should say, the lichens that grow on trees, when you see them, don't harm the tree. They're just occupying space that is there, particularly during the wetter months when there are no leaves on the tree. They don't harm the tree, so you should encourage lichens. Lichens are not mosses nor liverworts. You might have heard those terms. Those are plants, lower plants, that sometimes live with lichens, very often do. And they're usually green, like these two pictures here. Lichens usually have other colors than green. Lichens are named from the mycobiont, the fungal partner. It's just by international agreement. The fungi involved are unique to the lichen. They don't occur anywhere else. They're not free living. They reproduce in a variety of ways we'll talk about. The photobionts, the algae or the cyanobacteria, are there. They have names, but they're secondary to the name of the fungus. They are not known to reproduce sexually. They may, or they pretty obviously break up into pieces, so they reproduce asexually. Lichens are, the colors of lichens are distinguished from the dry color. Now here's a lichen. We didn't see any of this on any of your trees. I would hope that maybe somewhere on Whidbey Island there is some of this growing on oaks, but I don't know for sure. It's a common occurrence down in the Willamette Valley, but that's a little wetter down there. In any case, this lungwort is a large foliose lichen, and I thought I'd just show you this dry and then wet, a much closer view. And when you cross-section a lichen, you see that it's made up of several parts. In the case of this lobaria, it actually has 3 definite partners and probably more if you start looking real closely. But we'll get into some of the more recent studies of all the other partners in lichens in a bit. Here is the fungal part makes up this entire bit here with the green algae in a layer underneath. Now this is wet, so some pigment, not only the green algae, but pigment in the upper surface of the lichen has turned this to be bright green. Like I say, this is the color you would recognize it as or identify it as. Color is important in identification. This lichen also has blue-green bacteria and little clusters scattered around. And that's important ecologically. We'll get to that in a second. It also reproduces asexually. We'll get to that in a second. Here's an even closer view of a cross-section of one of the most common lichens that I saw down there, this one, Parmelia sulcata. And you can see that there's kind of a loose fungal layer, loose fungal layer, compact fungal layer at the top with the algae, the photosynthetic bit, in a layer right under the upper surface. This mimics the structures of leaves, actually. So the algae produce food by photosynthesis. The fungi are dependent on that food, but they provide the nice environment for the algae. So it is kind of a this for that, a mutualism. Some people say the algae are being parasitized, and in some ways they are, but they like to live in there because it's a nice place to live. So I think all in all, I like to call it mutualism. A little bit about reproduction. And let's compare reproduction of the lichen fungus with its free-living relatives, free-living fungi. Free-living fungi like mushrooms or this cup fungus, which is related to most lichens, produce spores periodically, but all at once in a fruiting body like these cups. This one comes out in the winter, probably late December through February. You might see this. So if we were to look at the insides of these cups, we'd see all kinds of spores produced all at once. And the cups last a couple of weeks at most. In the case of the lichens have similar fruiting bodies, many of them do, that produce spores. If we look inside them, there are just a few spores produced every once in a while. So these fruiting bodies last for maybe a year or longer and produce spores a few at a time. This is a big difference between the two groups. Now, as you might imagine, something that has two partners or more is going to have a hard time if each one of them has to reproduce, and then the partners get back together, the reproductive parts, the spores. So what lichens often do is reproduce asexually by budding. And there are two ways that there are a lot of different ways, but two basic ways that people have defined that lichens reproduce asexually. One is to produce these little flowery, F-L-O-U-R-Y structures called Rhoades. And this lichen that's so common down there does this. It reproduces both partners in these little structures here, the Rhoades. And if you were to look at one Rhoadium, one of these little powdery things here, this is what you'd see microscopically. The algal cells with a few of the fungal strands mixed up with it. So this structure can form by growth this thallus on its own. And that's a very handy way if you're two organisms growing as one, or perhaps three or more. Cerida and other asexual structures are blown about in the wind. They splash around with rain, and animals pick them up on their feet and carry them about. So that's a good way to reproduce. Another asexual form is Isidia. These differ from Cerida in having a hard outer rind over them. They are microscopic though. Here's another Parmelia, not one that we saw, but could easily occur, that has these Isidia on it. And here's a close-up of that. They're little wart-like structures. How do you identify lichens? Well, the growth form of the thallus. By the way, if you have any questions, shout them out. As I know you're all probably not used to looking at things like this, and if you want to know anything that I haven't mentioned, please ask. Anyway, the growth form, the color, as I said, is important. Whether they're reproductive structures, such as I've described, other kinds of structures, rhizines are root-like structures on the lower surface, where they grow. So with Garry Oak, of course, we'd be interested in those things that grow on deciduous trees, and the chemistry. Lichens are one of the groups of organisms where chemistry has played a role right from the beginning. And I don't want to get involved. I mean, here's a situation with this group of orange lichens. When you put some KOH on it, it turns purple because of a chemical that's in there. And a number of other chemicals occur in lichens. They can be separated out by chromatography, crystallized out, or use chemical tests to test for them. So it's extremely important when you get into lichen identification to use some of this chemistry. That maybe makes it a little difficult, but it's not that big a deal. Even if you don't have the chemistry, you can go a long way with it. Here are some of the different colors that lichens take. And you can see there's no real grass green colors. Mm-hmm. They're black, yellow, white, mm-hmm. kind of gray or olive color, but not grass green when dry. A couple of other odd sorts of things. This first one definitely would occur on the Garry Oak. You would find this situation. This is a situation where two lichens share a photobiont. They have the same algal partner. And so one, the orange one, can steal the algal partner from another lichen that shares it. Oops. So this is the polycoliona here. And it is stealing the algae from this Physia, which is a totally different color and really different growth form as well. You can sort of see that happening over here, where the lobes of the Physia thallus are beginning to turn orange because the fungus, which relates to that same alga, has started taking over this part of the thallus. Real odd sort of thing. One photobiont, two lichens. They're different species because they're different fungi.
Fred, eventually will that stunt the growth of the original lichen, or will they kind of alternate between sharing, or what usually happens? Does one win?
I think what happens is it looks to me like the polycoliona, we used to call this Xanthoria polycarp, basically steals the algae from the Physia and then takes off on its own. And whether the Physia can steal it back, I don't know. I don't really see that happening anywhere. There's a few of the twigs that I brought home from the Garry Oak, I thought I could see this beginning to happen. So you might go out and see if you can see it. But I think what happens is this lichen becomes independent of this one. In other words, they don't live together like this very long. There's another thing that happens occasionally, not usually on trees, but I just throw this in as an odd thing that you might see on the ground in certain environments. This picture was taken over in Friday Harbor, but I would think maybe it could happen around the oaks, but on the ground. This is a situation where there's one mycobiont, but two lichens, or at least two forms of the same lichen. Because they have the same mycobiont, they get the same name, which is Peltigera britannica. But in one case, the blue-green bacterium dominates. That's this dark, kind of slate-colored lichen. And it has little buds that have the green alga in it. And the other form is where the green alga predominates and the little buds have the blue-green in them. Very, very strange thing. But it is known to occur around the world. And this does occur locally here. One mycobiont, two lichens. Not to add confusion to things, but that's a situation that happens. And about 4 years ago, Tony Spribble discovered, when he was looking at some lichens, that there was another fungus in the lichen. And depending on how much of that other fungus, it was a yeast related to mushrooms. And you can see in these two boxes here, staining for that yeast in a microscopic preparation of these two forms of the same lichen. So in one case, when there was a lot of the yeast, the lichen took this form. And we used to call this Bryoria tortuosa, but it's now known that the fungus that gives it its name is the same as this lichen, Bryoria fremontii. So the correct name for that is Bryoria fremontii. And it has more of this yeast than this other form. And this was kind of an earth-shattering discovery that there were other fungi associated with lichens that seemed to be always present, but not necessarily always present in the same amount. And it got these people to looking, and they've found these yeasts in a great many species of lichens. So this puts a real monkey wrench into the whole naming, because the law says that the fungus gives the name. And since they're two fungi, what name? Well, for now, they're not worrying too much about this, but as this gets studied more and more, I think there'll be some more come out about this. For those of us in the lichen world, it was a major discovery, akin to Darwin discovering evolution by natural selection. Very, very interesting happenstance. One other thing, and then I'll talk a little bit about ecology and human importance. Anybody who's been involved with identifying things using keys, flowers, trees, ferns, animals, birds, has realized that recently with DNA study, things have become very messy. So I just wanted to give a visual as to how messy things are. So we have this family, and this is actually the DNA tree upon which the mess that I'm about to show you is based. But traditionally, this family, the Teleschistaceae, I mentioned earlier that a drop of KOH turns purple on everything in this family because they all have parietin in them. In any case, traditionally there were 3 genera. There was the Foliose genus, then Xanthoria. There was the Crustose genus, Caloplaca, and there was a Fruticose genus, Teleschistes. These are species that we have locally. Many of them are marine. We don't have any Teleschistes locally, so I didn't include it. So anyway, it was really nice if you could tell these genera apart by growth form. But when we looked at the DNA, this is what happened. So I'm going to rearrange these species according to the new taxonomy, which is exhibited over here. It's a mess.
Mm-hmm.
So there are all these new genera. And this is because things that were closely related or seemed to be closely related morphologically by growth form weren't closely related by DNA. So people have invented all these new names. And you can see Caloplaca is down here. Xanthoria is up here. But those were the 2 genera before. There are all these other genera. That's just a visual, I'll just do that one more time, a visual indication of the mess that looking at DNA has done with all our taxonomic names. It's why people think that common names might not work out better. Unfortunately, we don't have a lot of common names for lichens, but for birds and things, it works out better. Anyway, let's move on to ecology. We hear that soil formation is important. This is not an important feature for Garry Oak lichens, but I'll just talk about lichens in general here. It is true that lichens occupy bare rocks, and in doing so, they form a foothold on the rocks that helps break up the rock, mostly physically, not chemically. As they break up the rock, then they provide a foothold for more complex lichens and for bryophytes, mosses, and other bryophytes. And those things with 3-dimensional structure trap soil and provide a place for plants which have roots to take place. So that's a very slow process. It takes hundreds of years from a bare rock, but it is an important thing that lichens do. Any lichen that has a blue-green bacterium in it is potentially capable of fixing nitrogen. Nitrogen fixation takes atmospheric nitrogen, which is a gas, N2, and converts it into organic nitrogen. There are a number of lichens that have blue-greens. Some grow on Garry Oaks, not this peltigera here, but another peltigera I'll show you in a second. They have blue-green bacteria, at least part of their photosynthetic partners, so they can fix nitrogen. This process is the only way to get a large amount of organic nitrogen out of the air. Inorganic nitrogen, N2, is totally unreactive. We just breathe it in and out. The only reason it ever becomes important is when you dive and the N2 bubbles out of your blood. If you've dived deeply, you get the bends if you come up too fast. Not a major importance for tree growing lichens, but some lichens are eaten as food. Most famously, reindeer lichen is eaten in the winter by reindeer and caribou. They scrape away the snow and eat it. It doesn't have a whole lot in it, but it's enough to at least fill up their bellies and make them think that they've eaten. It does have a little bit of protein. And there's probably a few lichens that are actually eaten by humans, not in this country particularly. Not an important foodstuff for human beings. Mostly, if you ate lichen, you'd be eating leather. It's very tough and hard to digest. Slugs like lichens. I think they mostly like the algae that are in lichens. So you'll often see on trees the upper surface scraped away to expose the areas that the slugs have eaten to get the algae. Some birds use lichens and mosses actually also for nesting material. And almost any lichen or moss, if you incubate it in water overnight, you'll get these little water bears. These are real tiny. You can see, you know, that from there to there would be 1/10 of a millimeter. So these are a fraction of a millimeter big, but they are kind of cute animals. And if you've ever heard anything about tardigrades, you've heard how resistant they are to drying. People have taken them up into space, exposed them to the vacuum of space, and then brought them back inside, put them in some water, and then they crawl around again. They're quite amazing little animals, and they're pretty much present in every lichen that you could scrape off of or moss for that matter, of a Garry Oak. Interesting little animals. Might be a fun thing for a high school project to look at these.
Fred, are those tardigrades eating? Are they eating the algae, or what are they eating?
I think if they're eating anything, it's probably the bacteria that are in and around lichens and other little bits of detritus. I don't think they don't really have the means to get into lichens to eat them. So if there's some algae that break out, sure, they would eat them. So maybe some of those Cerediia. Looks like this was from a moss. I'm not sure that looks a little green there. This would be the tardigrade stomach here. I don't know what they've eaten. That'd be an interesting thing to do. I mean, it's not impossible to isolate these. Like I say, if you just take lichen or moss, soak them in water, distilled water overnight, and then look in the bottom of the jar, you'll find these crawling around. So they're not hard to find. They're just small, and I would think this would be a great high school project to look for tardigrades and then see what they eat. Probably somebody's discovered what they eat. I just don't know it. Lichens, because they're like sponges, are useful in monitoring air quality. Now, there's two ways to do this. One is to look for sensitive species and compare an inventory, where actually you look for non-sensitive species. You look for all the species, but you can arrange the species by sensitivity. This was first done in the 1970s in England. And then my mentor at OSU, Bill Dennison, and his students down there back in the 70s did a similar thing on Garry Oak and Oregon ash in the Willamette Valley. And they were able to map air quality. The darker areas are more urban areas, as you can see. I mean, here's Portland, Salem, I think that's Corvallis, Albany, Eugene, So they're quite sensitive. And the way they would do this is they look on the Garry Oak at 5 and 10-year-old twigs. You can age oak twigs by looking for the bud scars. And so here's the terminal bud, which will grow into the next year's branch. This was last year's terminal bud. And you can see the bud scars going back. So this down here is a 5-year-old twig segment. So,
I knew that.
pardon?
I said I never knew that. I find that very interesting.
Yeah, take a look. They're not hard to see. So you can age a twig quite easily from the bud scars. So what they did is they looked at lichens just on 5-year and just 10-year twigs. And this eliminates some of the variability. That's why they did that. And then they have a form, and this is in a book which is no longer available, but there is a link on the internet that you can download. I can send you this. I'll send it to Laura, and then she can post it or whatever. It's in my list of links on lichens. In any case, I just looked at their little scoring sheet here, and the Garry Oaks in Oak Harbor have at least these lichens. I did not see any Loberia pulmonaria, nor any of this species of Physia. So they're just, that's very simple. You know, I didn't do it exactingly like they want you to do it, but that would be typical rural air. Very cleanest air would have Loberia pulmonaria and or Ipolia. Of course, this is down in Willamette Valley. You could do something like this on Whidbey Island. We did something similar up in Bellingham, but we were using non-oak habitats. So it is definitely possible to do this kind of thing. The other thing about lichens as monitors of air quality is if you look at the most resistant ones, the ones that tolerate air pollution the most, and then measure the amount of air pollutants, heavy metals and sulfur particularly, you can map out the levels of these pollutants. So this is some work I did over in the Olympics back in the late 80s. And this is mapping lead according from the distance from the highway. This was just after leaded gas had been eliminated from our gas supply. So there was still a signature there of lead. So this is, actually, this would be the edge of the highway there. This is running up into the park from Port Angeles. And this is moving away in meters away from the highway. You see how the lead level drops off. Similarly, sulfur dropped off from sulfur output from a paper mill in Port Angeles that no longer is there as you moved up into the park. So this is miles from Port Angeles up the road into the park. Interesting thing you can do with lichens. They're sponges. They soak up stuff to make their lives. And if there happens to be air pollutants in there, they soak that up too. Melissa can show you some of her yarns that have been dyed with lichens, but there are a number of very beautiful colors you can get from lichens. If you ferment lichen with ammonia, you get some beautiful magenta colors. Just natural dyes can be yellows and browns and some greens. Beautiful colors. And to something you can can do. This is a plate from a book from Alaska, but plenty of the lichens around you can be used to dye yarn. And they don't need mordants. Mordants are heavy metal glue, basically, that helps the dye molecule stick to the fiber.
Mhm.
Lichen dyes are self-mordanting.
Wow, I never knew that either. Huh. That's
Um,
really cool. I was gonna ask about the use of dyes, specifically like the Native Americans using them to dye maybe baskets or some special textile.
yeah, I have not heard of Native Americans using lichens as dyes. Melissa, have you?
Actually, they were extensively used. Like, for example, the Chilcot blanket of the Tlingit was made with a lichen dye. And also the red alder, the bark from the alder was used to make the red dye combined with the cedar to turn it red. And there's a black dye made from the skunk cabbage, and from also the black soil was used as a dye, like the black mud was used to dye cedar black as well. So dyes were made from a variety of sources and used by the first people.
I'm just thinking you'd have to pick yourself a whole ton of lichen in order to.
This is true. It
I
takes
know.
about an equal amount of lichen and yarn.
Yeah, that's a lot. What about medicinally?
That's true, and I didn't put anything in here about medicinal use. Usnic acid and perhaps some other lichen compounds have antiviral, antibacterial properties. The homeopathy uses usnic acid as a medicine, but the problem with homeopathy is that the dilution factors are so great, there's not likely to be any actual substance in the tinctures that they sell. I don't know if any of you are homeopaths, but they talk about the memory, the shadow memory that water takes of active principles like usnic acid. But if you look at the dilution factor for the usnic tinctures that homeopathic medicines use, they're very high, in the millions, so that the chance of there actually being anything in there is little. But the molecules are known to have medicinal effects. One other thing, again, not something you're likely going to do with Garry Oaks, but lichens can be used to measure age because they grow at a known rate. So we were doing some things up on a cairn in near Sitka that the boat owner that took me up there felt might be left over from 300 years ago when the Russians were investigating the area. The problem is this lichen, which Parmelia saxatilis, grows so fast. This is about life size on my screen. Just in 3 years, it grew that much. So the chances of it being useful to age something 300 years is very difficult. So I was not able to conclusively say, and there were no other crustose lichens which grow much slower on this cairn to use. So basically what you have to do is have a lichen that you know that you can measure the growth rate on, say, cemetery stones, where you know the age of them. And then you look for things that are of unknown age, and you can guess at the age from the size of the lichens on them. It's an interesting technique. It's called lichenometry. It's been used a little bit to date glacial moraines. Well, let me talk a little bit about the lichens that, and I've got quite a few pictures here, but I'll go through them quickly. Lichens on Garry Oaks. Now, this is what we see down in Oregon, which is, as I say, a moisture place, a little richer lichen availability down there. This paper by Villela, John Villela, and others, which you can download, has all the lichens that they found in Western Washington on Garry Oak. And to a certain extent, what I'm going to tell you in the following slides is based on what they found, but also what I saw when I was down with Scott and Melissa, and just my general knowledge of what is likely to occur. But pretty much everything I'm going to show you, Villela and others found somewhere in Washington on Garry Oak. Not that I suspect it is necessarily going to occur in Oak Harbor habitats. However, it should give you an idea of how many lichens there are. And otherwise, don't get bored by all the names, which I'll stop and talk about a couple of things, but mainly I'll just show you a bunch of pretty pictures. So we'll divide this up between lichens on the trunks of trees, on the branches of trees, and elsewhere, maybe on the rocks around or closer to marine habitats. So on trunks, one lichen that it stands out is this orange lichen, Chrysothrix candelaris.
Are you ready?
Yeah.
Oh, Keith. It's a powdery yellow-orange lichen, and you'll see it on many trees around, including the Garry Oaks.
Mm-hmm.
Another powdery lichen is this Lepra amara. It used to be called Pertusaria amara. It has little ceridiate patches, and it's a crust. And you might wonder, amara means bitter. If you put your finger on it and then taste it, it's very, very bitter. In the background there was a picture on Garry Oak. This is an interesting lichen that I found on Lopez, actually on the rare and endangered lichen list, Pyrospora quernea. It's a little powdery lichen with red fruiting bodies. And it's actually not uncommon on your Garry Oaks. Just to give you a flavor of what's involved in looking at crusts, this was something I'd never seen before. It was on your one of your Garry Oaks, and it's this little thing that has cups. It was a gray day, and I was shaking, so this is sort of out of focus, but this is called Cresponia chloroconia. I'd never seen this thing before, but if you section one of these cups, you see something like this. And then if you look close, it has these spore-producing cells called asci. And if you look in the ascus, there's a spore. They're four-celled spores, look like little clubs. And that is this genus, this species. It's known, actually, I don't think Villela found it anywhere else in Washington, but there it is on your oaks. It's not an uncommon thing around in Western Washington, apparently, but I've never seen it before. You can find this lichen pretty much anywhere. Here it is on the lower trunk. This is Ramalina farinacea. It's a fruticose lichen. And if you look at it very closely, you see it has these containers of Cerida dotting along the edges of the lobes. It's called the dotted line. It has a common name, the dotted line. Another lichen that looks almost identical to it is oak moss or staghorn moss, but it's actually a lichen, staghorn lichen, I guess we'll call it. There, the Cerida are on the surface, not on the edges. This is Evernia prunastri. Probably Gramolina is more common in your area, but they're both there and easily mistaken for one another. This genus of lichens is extremely common everywhere, particularly trunks and the bases of trees, but also on the ground. And I just threw in a whole bunch of them here. They're all in this genus Cladonia. They have little scaly pattern on the substrate, and then they put up some kind of upright portion. Here's another one. Here's another one. Extremely common genus, Cladonia. And you'll see these around in a variety of habitats, not just on tree trunks. One of the peltigeras that can fix nitrogen, I didn't actually see this. It's probably quite sensitive to air pollution. You probably would have to get out of town to see it. Peltigera colina. It grows on tree trunks and it has Cerida on the edges. The peltigeras have these little root-like things on the bottom, and they're called the vein lichens. This is an unusual thing I hadn't seen before. It was on the Garry Oak on Fidalgo Street there in the middle of town. Disconia enteroxantha. Moving to branches, there's a lot of Ramolina and Evernia, plus lots of other things on branches. Here are some of them. Usneas are an important branch genus. Usneas are a fruticose genus that if you look at them closely, have a resistant central cord down the middle. They're usually round in cross-section, have a variety of branches. They're quite difficult to get to species, but they are very common on your oaks. Here's one, the fishbone usnea that sort of looks like it has these fish bones on the side, Usnea dasypoga. This is one I have not seen down there, but if anybody sees this, let me know, because it would be the furthest south that anyone has ever seen it. But it seems to be moving south. It's the only usnea in our area that has the fruiting structure of the fungus on it, the apothecium. They're very pretty lichens, and it's quite common in Bellingham now. I've seen it at Washington Park in great abundance and on the Swinomish Indian reservation, but I've not seen it as far south as Oak Harbor. Keep your eyes peeled for this one. Some other foliose lichens, like I say, Parmelia sulcata, very common, probably the most common lichen on your trees. There's another Parmelia that looks almost identical, but it has isidia on the lobes, and not just restricted to ridges. It's called Parmelia hygrophila. That's there. This is a kind of a greenish, olive greenish Parmelia-like thing that's present. This hypogymneas are hollow lichens. This one has Cerida on the edges of the lobes. These little tiny lichens, this lichen here and this Polycoliona polycarpa where the ones I was showing you earlier share the same alga. And I think this may have the same alga also. These are two different physias, very tiny lichens on branches. I didn't see any of this, but it's the most common lichen in Western Washington. So it's probably around Plasmatia glauca, known as ragbag. Very, very common. This is quite common elsewhere, but I didn't see any of it. Actually, I did see this. Trichomonopsis chlorophylla. It's a brown lichen. Things that fix nitrogen. This other lobaria that is related to lungwort might occur. It has been found elsewhere on Garry Oak by John Villella. This is a blue-green bacterium containing lichen. These things are about an inch across. Sorry, I don't have scales and all these things. It's an interesting one. It smells like fish when you scrape it. But it could be present in some of the cleaner air areas. And finally, elsewhere. I put this in because I don't have any pictures of oak stands in these coastal bluffs, but I know they occur. This is actually a wind-blown Douglas fir over on Point Colville. But similar habitats occur with oak in some places in Washington. I don't know if you know of any in particular, but where there's marine influence, you might find some of these other things. Actually, this you would find on rock anywhere. This is this Parmelia saxatilis, grows on rock, has isidia. These two lichens grow on the ground. This one is a gelatinous lichen, built a little differently than other lichens. It has blue-green bacteria as its sole photobiont. And there are some of these that grow on trunks on Garry Oak, elsewhere in Washington. This is Leptogium palmatum. This other thing is the mushroom lichen. There are few relatives of mushrooms that form lichens. This is its thallus here. These occur on the ground or on rotten wood in coastal bluffs. This Hypogymnia is a marine species that would occur on Garry Oak next to the marine influence. And this very interesting Ramalina. Now, Scott and Melissa and I saw this. It was actually growing on a pear tree down south in that naval base property. Forgotten what that point was called. But this is the fishnet lichen, Ramalina menziesii. Very unique lichen. It's the only one in the world that grows like this, as far as I know. And it occurs in areas close to salt water in our area. As does this Bryoria fremontii. This is the one that has an extra amount of the fungal yeast in it that used to be called Bryoria tortuosa. You might see this on Garry Oak, again, closer to salt water. And you might find in those coastal bluffs these reindeer-like lichens in the genus Cladonia. These aren't reindeer lichen themselves, they're related, however. And that is the end of my presentation.
Well, cool. Thank you.
So I hope there's still some people watching.
Oh yeah. I learned a lot. I really, I mean, it's amazing how they're these small, silent, beautifully intricate, complex life forms all around us, and we don't even really think about. I mean, we just pass them by. My background is nursing, and I couldn't help but wonder if there's some cure for some diseases that we continue to battle. If only we could find them in the lichen.
Well, that's a good reason for that. I mean, that's one good reason for learning about other organisms and keeping these organisms around and keeping the habitats that they occur in. One thing I should say, you know, you're trying to encourage regrowth of Garry Oak. You don't have to worry about the lichens. They will come in. They will find the oak. So although you could potentially plant them, you don't really need to. They will find it and set up shop. So I'm encouraged that you're adding to the lichen habitat by encouraging the Garry Oak.
Mm-hmm. Yeah.
But it's unfortunate that people don't learn about the great diversity of these kinds of life forms that are around us, particularly in the Northwest, where they're so common and abundant, you know, really. Same could be said for bryophytes, the mosses and liverworts.
Mm-hmm. Yeah. Well, cool. One quick question. You at the beginning of the portion where you showed photos of the different lichens, there was one, I think it was the second one you showed, and you said it was it was actually an endangered lichen.
Yeah, it's I don't have my list here.
Okay.
Pyrospora quernea is its name, and it's Pyrrhospora Quernea.
Okay.
It's one of those things that probably is more common than is realized. But when they make these rare and endangered lichen lists, they have only to go on what we know, and there just aren't enough lichenologists around to say how widespread some of them are. But I was surprised to see it there.
Yeah.
And it's in the middle of town. So
Yeah.
it apparently is not, you know, whatever it is that, and maybe there isn't that much pollution in the middle of Oak Harbor. I don't know. But it seems to be very happy there.
Yeah. Well, thank you again so much. Does anyone have, so what time is it now? It's almost 8 o'clock. I'd like to wrap it up. Does anyone have any questions for Dr. Fred before we say goodbye?
I do have a question. Is there any kind of a interaction between the lichens and the trees that they grow on typically, particularly the oak trees?
They're occupying space basically between the photosynthetic leaves. If a tree dies for some other reason, the lichens will flourish there sometimes. And that's why people think that it's the lichens' fault, but they're just occupying space. They don't actually do anything to the tree.
Does the tree benefit in some way?
Well, in as much as the lichens harbor perhaps beneficial insects or hold on to water a little bit. That one of the ecological roles of lichens I didn't mention was as sponges holding water in environments. So they increase the water holding potential of the bark that they're on. But it's possible there are other benefits. If they're fixing nitrogen, they're supplying nitrogen to the system.
Mm-hmm.
And I should say there is one lichenologists, when asked this question, always say, well, they're not doing anything. But there's this one piece of work, which I've never seen, but somebody has told me about. And I see Katie Beck is with us.
Hello.
Maybe she's heard this also. In South America, someone traced Evernia prunastri, I think it was, or one of the Evernias, fungal threads down into the bark of the tree it was growing on and into the phloem of the tree, which would suggest that that was getting some benefit from, was perhaps parasitizing the tree. But I have never seen the study. I would like to see it. I just never figured out a way to find it. So I don't know if it's true or not.
Mhm. Yeah.
Okay, thank you.
Okay. Well, thanks again so much. It's really been a pleasure.
Anytime.
Yeah, you have a really easy teaching style. And so again, we thank you so much.